A warm welcome to everyone gathered here. In this video, we are going to see the demonstration of our project which is related to machine learning domain being used in agricultural sectors. Our project title is AgriDetect, which is a machine learning approach for timely plant disease identification using leaves. Myself, Maharaja K, and my team members are Meenakshi G, Prakash S, and Tarunika K. Our guide for this project is Professor Dr. V. Suresh Kumar. Okay, now let's have a look at the problem statement. In the existing systems, leaf diseases pose a significant challenge in jeopardizing both agricultural productivity and global food security. Although they have used only a single approach to identify the diseases in the leaves, that is, only a single algorithm is used for the identification of diseases, and this includes the usage of a limited number of datasets. Based on our research, we have found that comparative analysis would be useful for these types of systems. So we have designed our system in such a way that we made a comparison of four algorithms individually, which are support optimization, convolutional network, k nearest neighbor, and decision tree, to identify a specific disease of a specific leaf. And these are the datasets which we have gathered for our analysis. It consists of 5 different leaf datasets, which are apple leaves, grapes leaves, mango leaves, tomato leaves, and potato leaves. These leaves datasets comprises of various diseases within them. On the whole, we have gathered around 14,676 images for our analysis. So this is a proposed system. It consists of 7 stages. Initially, at the first stage, during data collection, the leaves inside the leaf datasets would be organized as separate folders, that is, healthy, diseased, and mixed. Here, the mixed folder will be given as the input to the system. This will undergo a series of processes such as image pre-processing using the normalization and image resizing methods, followed up by image segmentation based on the pixel intensities of the image. Then the feature extraction method is done based on shape, color, and texture of the leaf, in which we have used 4 different methods based on the machine learning algorithms used, that are color histogram method for support optimization algorithm, stochastic gradient descent momentum for convolutional neural network algorithm, and local binary pattern method for k nearest neighbor algorithm, and finally the histogram of gradients for the decision tree algorithm. After this, we do the model training and evaluation to predict the disease percentage in the mixed folder using multiple machine learning algorithms. This leads to the final stage of model fine-tuning, which helps us to find the best approach. Based on a system, we can conclude that which particular algorithm will be suitable for specific disease of a specific leaf. This type of approach will be suitable and efficient and also time consistent in order to predict the disease percentage optimally and spreading of the diseases at an early stage for sustainable farming. Now let us move on to the demonstration of a system. For our analysis, we have used MATLAB, a programming and numeric computing platform used by millions of engineers and scientists to analyze data, develop algorithms, and create models. I am using the R2024A version of MATLAB for this demonstration. This is the user interface of MATLAB. Here's the command window, and this is the current folder in which we are working. Here the details of the file selected will be displayed. Then this is the workspace in which all the variables and values of the models built could be monitored. MATLAB has various ROMs which are very much useful for making machine learning models at the easiest way. We have particularly used the image processing toolbox, computer vision toolbox, deep learning toolbox, and the statistics and machine learning toolbox for our analysis. Also, we have got the academic license for MATLAB given to us by our college, which was very useful for the development of a system. So let's begin. We have built a MATLAB app using the MATLAB app designer. Here in the app section, if we click the design app, the MATLAB app designer window will open. Here, this is our MATLAB app which we have developed, and this is the design view of the MATLAB app. It consists of 2 buttons, 2 drop-down menus, and 4 labels. Now let us run our MATLAB app by clicking the run button. Once I click the run button, the MATLAB app will be executed and the MATLAB app will be displayed like this. Here, there is a button called the select dataset folder button. I will be clicking this button. Now I have been asked to select the dataset folder which I want to test. I will be selecting the grapes leaves here by selecting the grapes leaves folder and clicking the select folder button. Then below this there is a label which displays the dataset being selected. Since I have selected the dataset as grapes leaves, the dataset selected is being shown here as grapes leaves. Now let us choose the disease type which we want to predict using the select disease type drop-down menu. There are 3 diseases available here because there are 3 subfolders inside the grapes leaves folder. Now I am choosing the grapes black rot as the disease that I want to choose for this grapes leaves dataset. So after selecting the disease selected is displayed as grapes black rot in the disease selected label below. Then I would be choosing the algorithm with the help of the other drop-down menu below and choosing the SPM algorithm as my choice to perform. Once I select the algorithm selected will be displayed here as SPM algorithm in the label as I chosen it. Finally, I will be clicking the start prediction button. It will take around 1 to 2 minutes for the output to be displayed here. In the meantime, I will explain what's actually happening here. This system now will be performing the SVM algorithm for the grapes leaves based on the grapes black rot disease and will return the disease percentage of the leaves in the mixed folder. As we have organized the folders with the healthy, diseased and mixed leaves, we will be knowing the actual percentage of the diseased leaves and now the output is displayed. So our predicted percentage here is 52.73% of diseased leaves, whereas the actual percentage of diseased leaves here is 52.74. So SVM must perform good for this approach. Like this, we have done all the 4 algorithms for each and every dataset with its disease type individually and made analysis through it. Here are the results of the analysis compared to the actual result and the predicted result. These are the disease percentages which we have manually calculated with help of the number of disease leaves and the healthy leaves. This is done by combining both the healthy and disease leaves of a particular dataset and storing it in the mixed folder. Then, dividing the total number of disease leaves with the number of leaf images in the mixed folder will give the actual disease percentage as a result. So, with the help of those actual percentages, we have comparatively done a machine learning algorithm approaches and found out that which algorithm would be suitable for the specific disease of a specific leaf dataset. Mostly, convolutional neural network algorithm and the support vector machine algorithm is matching up with the actual results. With this, I complete this video. So those who have watched this video, thanks for watching. Don't forget to like, share and subscribe. Once again, thank you all.